Howdy, Earth historians. Welcome to another Earth History online post. The subject today is photosynthesis, the process that plants and related microbes use to obtain energy from sunlight and synthesize organic matter from nothing more than carbon dioxide and water. Second to the actual origin of life, photosynthesis is perhaps the single most influential evolutionary innovation in the history of the Earth life system. Not only did photosynthesis make it possible to greatly expand the total amount of life on Earth by producing organic matter, it also began pumping oxygen into the atmosphere. And oxygen is a very reactive compound. It's poisonous to many forms of microbial life and reacts with inorganic reduced compounds in the oceans and the atmosphere. I'll explain some of the consequences of this in class. The aim of this post is to provide you with an understanding of how oxygenic, that is to say, oxygen-producing photosynthesis, how it works, and why it may have taken so long to appear after the origin of life. The basic chemical reaction that summarizes oxygenic photosynthesis is shown right here on the upper right-hand side of the slide. It says carbon dioxide plus water plus light yields organic matter and oxygen. But this simple summary hides a huge number of complex steps. Let's start off with how biologists divide the oxygenic photosynthesis pathway. They think of photosynthesis in terms of two parts, the light reactions and the dark reactions. Let me start with the dark reactions. These are actually familiar to you. It's basically a metabolic cycle. The cycle takes in hydrogen ions, carbon dioxide, and energy from ATP, that's there and here, and they produce sugar, which is an organic compound from those inorganic constituents. This should be familiar because it's much like the reverse citric acid cycle that we've already encountered. In this case, it's a different cycle called the Calvin cycle, but like the reverse citric acid cycle, it takes in carbon dioxide and hydrogen and energy and ends up producing new organic matter. This isn't new, and for our purposes, it's really all you need to know in terms of the details of the dark reaction of photosynthesis. This is the cycle that produces the organic matter. The really big innovation of photosynthesis is in the light reactions. These provide two critical materials that are required for the metabolic cycle in the dark reactions. They provide the hydrogen ions needed to create the organic matter, and they also provide energy in the form of ATP. Oxygenic photosynthesis requires three distinct steps. The first is down here. That's the water splitting step, and it breaks water into hydrogen ions and oxygen. And in fact, this is the source of oxygen that's the waste product of oxygenic photosynthesis. After the water is broken into hydrogen and oxygen, it actually takes two other steps called photosystem 2 and photosystem 1 to be able to produce enough energy and the raw materials that are necessary to drive the dark reactions. I'll point out that the numbers in terms of the order of these photosystems do not reflect when they occur during the light reactions. They reflect the order of discovery. Photosystem 1 was discovered before photosystem 2, even though it happens after photosystem 2 in the process of the light reactions. Okay, let's walk through each of these steps and understand the role of each of these steps in the light reactions to understand why it's necessary. The place to start is the microbial tower of power. Now recall that metabolisms obtain energy from paired reduction-oxidation reactions in which an electron is exchanged. If the oxidation reaction, shown here on the left-hand side, releases more energy than is necessary to drive a reduction reaction, shown here on the right-hand side, that surplus energy can be harvested metabolically. Such combinations are these downhill reactions, the orange line here, the yellow line here, and the red line here. These are summarized down here in terms of their chemistry on the lower left-hand side of the slide. But from a chemical standpoint, it's possible for a reduction reaction to require more energy than is released by an oxidation reaction. Such a pair can only run if energy is actually added to the system. Essentially, these are the equivalent of uphill reactions. For example, this blue reaction or this green reaction. Those two arrows are actually summarized down here on the lower right-hand side of the slide. The trick to making these kinds of reactions run is to be able to find some additional source of energy to push them uphill. This challenge is met by a group of organisms called the phototrophs or photoautotrophs. Phototroph translates as light-eating organisms. They obtain the energy to drive these redox reactions uphill energetically by harvesting energy from light. And the molecule that allows them to do this is chlorophyll. Chlorophyll is a complex protein that includes a ring-like structure that contains a magnesium ion. The energy in light is carried in subatomic particles called photons. The amount of energy contained in any particular photon actually corresponds to the color of light that we see. Infrared photons in light are lower energy, ultraviolet photons in light are high energy, and in between those two ranges is the visible spectrum of colors that we can see with our eyes. Now, the structure of chlorophyll with this magnesium ion trapped in this ring allows it to absorb a photon's energy and release an electron. Essentially, what's happening is that chlorophyll is oxidized when it's hit by light. That oxidation reaction can be paired with a reduction reaction that can in turn drive a metabolic chain. Note that by giving up an electron, chlorophyll is going to need to replace it, in other words, to be reduced, in order to be able to carry out this trick again. Let me show you how this works. On the earlier slide, where I showed a summary of all the reactions required for oxygenic photosynthesis, I pointed out that chlorophyll needs to capture a photon and increase energy in 2 steps. But for the moment, let's focus on these steps separately. Remarkably, each of these individual steps is carried out by different types of bacteria. These are actually carrying out photosynthesis, but it's not oxygenic photosynthesis. It doesn't release oxygen. We'll see how that is incorporated by oxygenic photosynthesizers as we continue with this posting. Now, in terms of the metabolic tower of power, here's how chlorophyll works. Let's look at this first cycle here on the left-hand side. Chlorophyll initially starts down here. This is reduced chlorophyll. It's already got an electron that it can give up. When the photon hits that chlorophyll, the energy level of the chlorophyll rises up to this level. This level can then drive a series of redox reactions from which energy, ATP, can be harvested. That's this series that goes downhill. Remember, each of these steps is a redox reaction. It's mediated by proteins and enzymes, and it only works because each reaction releases a little bit more energy than it takes in. That's why it flows downhill. In bacteria, there are 2 slightly different types of chlorophyll that each start at slightly different energy levels and absorb slightly different photons, slightly different colors of light. In some bacteria, the cycle is closed. That's shown here on the right-hand side. So here's this particular type of chlorophyll. It gets energized by a photon that allows it to release an electron through this chain of reactions through here. That electron is then used to replace the original electron that was given up by the chlorophyll up here at this stage. A closed cycle like this can be used to generate ATP, but it doesn't actually generate another critical ingredient that's needed to create organic matter, and that is hydrogen. You can contrast this to the cycle on the left. In this case, The chlorophyll starts down here, it gets energized by the photon here, that electron goes down here, and then it gets captured by this molecule called NAD. NAD uses the energy of the electron to transport hydrogen into the dark reactions. So if it takes away the electron, how do we recharge the chlorophyll? Well, we do this by taking a reduced material, in this case hydrogen sulfide, and obtaining an electron from it to replace the one that drifted off with NAD. The advantage of using a material like hydrogen sulfide is also that it can provide a source for the hydrogen that NAD is going to transfer into the dark reactions. If an unclosed chain reaction like this on the left is used, then sulfur ends up being produced as a waste product, and the electrons in hydrogen of the original hydrogen sulfide are consumed in the production of organic matter. Before I continue, I need to point out that each of these either cycles or the complete chain reaction that's described in these two diagrams is called a photosystem. The particular part of the photosystem that's highlighted either in green in photosystem 1 or purple in photosystem 2 is called a reaction center, and that's where electrons are transferred through a series of enzymes and proteins. These are just terms. You need to be familiar with them in order to be able to talk about this stuff. In any case, having just one of these photosystems or reaction centers works just fine for bacteria. So why do oxygenic photosynthesizers like plants need both? Let's take a look. Okay, so running a photosystem as a closed cycle generates energy, but it doesn't generate the hydrogen ions that are needed to synthesize organic matter. Using a compound like hydrogen sulfide to run an unclosed photosystem generates energy and hydrogen, but it requires a raw material, the hydrogen sulfide. Such bacteria that run photosystems like this must be in a location where hydrogen sulfide is therefore available. You know what compound is much more available on Earth and also has electrons and hydrogen to use? Water, H2O. But there's a difficulty with using water as the source of electrons needed to reduce chlorophyll. The problem is that oxidation of water into hydrogen and oxygen is really low on the metabolic tower of power, substantially lower than the oxidation of hydrogen sulfide. You can see the difference right here. Oxidation of water requires a special catalyst called the water splitting complex, and this is a very complicated protein with 4 manganese ions and a calcium. That's shown up here. You don't need to know the details of this structure, but you do need to remember that it involves the element manganese and that this isn't a trivially easy chemical reaction to manage. Now, if oxidation of water is going to be coupled with the reduction of chlorophyll, this requires that the oxidation reaction be higher on the microbial tower of power than the chlorophyll reduction. But if the reduced chlorophyll that results from this redox reaction is so low on this metabolic tower of power, how could it possibly run any further redox reactions? Okay, this slide compares the two photosystems I've shown you before, type 2 and type 1, with the combination of the two that's used in oxygenic photosynthesis in the middle. All right, now, remember, chlorophyll can absorb a photon of light, which allows it to shift to a higher position on the tower of power. That's all of these vertical up arrows. However, when using H2O, water, as the source of its electrons, it starts so low, so far down on this tower, that just one photon isn't enough to drive the metabolic cycle that's necessary to synthesize organic matter in the dark reactions. So the result is that chlorophyll has to raise up the energy of the transferred electron twice. The first time from water here to this level, that allows this cascade to proceed. The electron that results from this cascade starts here. It reduces a different chlorophyll molecule that then absorbs light and steps all the way up here, and it's only from here that this energy is sufficient for NAD to capture the hydrogen from the water here to be able to transfer it to the dark reactions and drive the whole system that synthesizes organic carbon. This system is like a Rube Goldberg machine. It's cobbled together to do something in a much more complicated way than we would expect if we were, say, designing this as engineers. The reason for this complicated two-part system, however, is that chlorophyll is simply limited in how much energy it can add to an electron. It simply can't raise the energy of the electron to drive the cascade any further than this. It simply can't do this extra step in just one reaction. That's just a limitation of the molecule. Okay, so here's my point in going through all this rigmarole and posting this explanation. For oxygenic photosynthesis to evolve on Earth as we see it today, it required the appearance of the photosystem cycles, the dark reaction cycles, and the water-splitting complex. As we'll see in class, each of these individually is actually not all that difficult, and there's evidence that each of them evolved much earlier in the history of life than the first time we see evidence for oxygen production. The evolutionary difficulty wasn't actually coming up with a series of metabolic cascades and cycles. We've seen that before. The challenge was how to link them together so that they could actually work consistently without interfering with one another. This appears to have been much more difficult. However, the payoff, the ability to create energy-rich organic matter from virtually unlimited materials like water and carbon dioxide, was a great evolutionary advantage for the lineage that invented it. The sheer amount of living and organic matter on Earth could explode, and the waste product that it released, oxygen, was both an opportunity. After all, the most efficient form of energy metabolism is aerobic respiration, which requires oxygen. It was also a catastrophe, because oxygen is a highly reactive toxin to living organisms composed of reduced organic compounds. Okay, that's what I would like you to be familiar with for class, so please be ready when I ask you about it. We'll go into the details about when there's evidence for the first emergence of some of these aspects of oxygenic photosynthesis and where they may have come from over the next day or two. I'll see you in class.